Papu is super ready for the experiment number 2. I hope you guys are also ready. Now today we are going to do the experiment on pollen tube growth. So let's see what all are the required things. So first of all, my dear children, we all know we always require vinca rosae flower. This is super important because we are going to take the stigma and style of this particular flower for the experiment. The pollen grains lands on the stigma and then forms the pollen tube, right? So which is why we need it. Now the second thing that we would definitely need is some hot water. So either in your institution they might provide you the hot water. I am not sure if at all you can see the fumes coming up, but it's there. Or there will be a burner and all the setups up there. You might have to heat the water and keep it ready, that's all. Now going ahead, you will definitely need a glass slide and also a cover slip. And cotton blue is the stain that we are going to use for viewing it under the microscope. And we would need a watch glass for putting in the stain, and we would need a brush to transfer the stigma from the watch glass to our glass slide as well. So, and last but not least, you would also require the microscope. As I always say, please view it under 10x power. So let's get ahead with this experiment. Okay, so the first thing that you need is vinca rosae. You can dissect it using a needle, and this is what we will get. This is the stigma as well as the style. Now we would be putting it inside some hot water so that the tissues become softened to take up the stain that we are going to use. So over here, with the help of a brush, transfer this onto a clean glass slide after some time, and at this point, if at all you want to cut off the extra portion, you can, and then we can add the cotton blue stain which is there using the brush or even a dropper. Further, it will look something like this. You need to put the cover slip on the top of it and then dab off the extra stain, and also make sure to press it gently and then view it under the microscope under 10x power. Ta-da! I hope you enjoyed the experiment. Well, my dear children, this is how it is going to look under the microscope. So these are your pollen grains and they have the pollen tube as well. And the color is because you used cotton blue stain. Now, as I always say, wherever you are studying, they might be following some other procedure to do this particular experiment. So please coordinate with the institutions wherever you study. The common things remain the same only, okay? And according to the lab manual which is given to you, the steps goes like this. So I thought of explaining this as well. If at all someone has any confusion that, ma'am, why did you do it like this in our lab manual? It's something else. Don't worry, everything is the same only. The end result is going to be the same. So listen to me carefully how it goes according to your lab manual. So here they have taken a beaker with water and cotton blue stain and they had boiled it. So water along with cotton blue stain has been put and they boiled it. Okay, we did it separately, they did it together. Now, place the cut styles with stigma in boiling water for 5 to 10 minutes to soften the tissue. So why do you need to put the stigma as well as the style portion into the water? Because you want to soften the tissue so that this particular cell which is there, or these cells which are there, they can take up the stain that you have mixed, which is what? Cotton blue. So in our video, we boiled it we put it inside the boiling water and then we took it and then we added cotton blue. But anyway, we did put in the hot water. Why? Because we wanted to make sure that the cells are now softened. They can take up the stain which we are going to put. Now, after that, what we did in the video is that we put a bit more stain and basically we put the cover slip properly and we viewed it. And before viewing it, basically we gave a press also, right? A gentle press was needed over there, and then basically we viewed it under the microscope. Now over here, some people, okay, they also put a drop of glycerin. So after taking this particular thing out from the mixture of water as well as cotton blue, so it has got the stain, okay. Now you take it out and you can put a drop of glycerin. Why is it being done? So glycerin prevents drying up, okay, it prevents drying up. So that is the only reason why we use glycerin over here. Some people do, some people do not. And then after putting the glycerin, you can put a cover slip and then gently, okay, gently press on the material. Don't do like this, otherwise the cover slip is going to break up. You would not be able to see anything, okay, you would have to repeat it again. Otherwise, you can also use the back side of the pencil and gently press it up with the help of that. Okay, and if there is any stain towards the side, please remove it with the help of blotting paper as well. Now after that, you can directly view it under the microscope, and as I always say, 10x. Okay, so one more line is written: if you fail to observe pollen tubes, mount another stigma, observe the slides under a microscope. So either way you have to observe it. Now one very important thing, the extra portion of the style you can cut it off as well. Okay, after putting it in the hot water, and when you take it on the slide, you can cut it off and then add glycerin, and whatever thing is be done, you can do that. Okay, similarly in the video that we did also, you could have cut the style portion, the extra portion, and then you can add the stain. That would be better. After putting the stain, if you go and cut, it might be a bit troublesome for you. Okay, so yes. Now one very important thing, you do not get marks just by doing this. By doing this, by preparation of the slide, you get 3 marks. But now still you need to have 2 more marks. Where do you get that from? Let's go ahead and check that. But before that, have you subscribed to the channel yet? If not, subscribe to it right away, my dear children, because we are doing so many things for you, right? So we deserve some form of love in the form of subscriptions as well. Okay, now coming over here, you need to also make diagram. When you make the diagram, you get those 2 marks, and this diagram needs certain labelings. Okay, certain labelings such as stigma, style, and the pollen grain, and also the pollen tube. So first of all, make the pollen grain and label it, make the pollen tube and label it, label the stigma, label the style. Each of them carries half marks, half marks. Okay, so half into 4 is equals to what? 2 marks. So you will get your 2 marks out there. So this is the experiment for pollen tube growth, and this is how you are going to get your 5 marks out of this. And my dear children, so with this we are done with the second question also. So as I always say, out of these 4 questions you might be given 1 to do. Okay, so we have already done it in the previous video. If you have not watched it, go watch it. And now in this particular video, we did the pollen tube growth experiment as well. Now the next video, we will be seeing the number of locules and the type of placentation in hibiscus or China rose. Also, if at all you are someone who is preparing for KCET, do not forget to enroll into ABHYAS KCET test series. It's going to be a really helpful test series for you because we have got 35 mock tests. There is chapter-wise KCET PYQs and all of it in exam-like condition. In 60 minutes, you have to solve it. And this is the most trusted test series as well. We had so many, rankers out there from this test series. So we recommend it to you. If you need it, definitely check it out in the link given in the description section below. Wishing you all the best, and that's all for today. See you soon with the next experiment.